All right, we are in section 2 of notes. We're talking about the diversity of fungi, so we're going to be talking about all the different phylas. All right, so we're going to talk about 4 major phyla of fungi. We're going to look at the distinguishing traits of each phylum, the reproductive strategies of each phylum, and then we're going to start talking about some lichens and mycorrhizal relationships, and then even beneficial and harmful fungi. All right. So that is the main goal of this section of notes. We do have some vocabulary down here to know, learn, put into your brain forever. All right, but let's jump into it. So when we're talking about classification of fungi, we're going to be looking specifically at 2 things, right? Specifically structure and methods of reproduction. All right, those are the 2 main things that are dividing our fungi into our 4 major phyla. Now there are 4. We have phylum Chytromycota, which is our chytrids. We have Zygomycota for our common molds, Ascomycota for our sac fungi, and Basidiomycota for club fungi. For fungi, it is going to be important to know the phylum names, right? So these are going to be important. But let's look at chytrids first. All right, chytrids are members of phylum Chytridmycota. All right, that's where the name comes from. And these are going to be saprophytes and parasites, right? So that means they are decomposing dead things and feeding off other things, right? So this means that they are heterotrophic, right? So they are going to be decomposing things or being parasites. The interesting thing is that most of these are going to be aquatic, so they're living in an aquatic environment. And because they're in water and in an aquatic environment, they actually produce flagellated spores. So their spores actually have flagella. We can see this over here in this picture because we do have this flagella that is right there on that spore. So we do have flagellated spores. So our spores do have flagella on them to help them navigate that aquatic environment. Also, because it's in an aquatic environment and we do have these spores, a lot of times people think that there's an evolutionary link between fungi, right, our chytrids, and our fungus-like protists, right? So chytrids are kind of considered the link between fungus-like protists and fungi. Alrighty, so we're in aquatic environments, right, and we're going to have some different things there. Common molds are the second phylum. They are in phylum Zygomycota. All right, and Zygomycota is mostly terrestrial, so they're mostly on land. Interestingly enough, we will have some that are in a mutualistic relationship with plants. We'll have some, not all. But a lot of what they do is going to be molds, right? Common molds that we think of is what we call them. And so because they are molds, they do have specialized hyphae, right? Remember, hyphae is that chain of cells that are linked together where we have that septa, all right, where we have the cross walls, so septa, right, to allow nutrients to move through. So we do have specialized hyphae, and they're called stolons. So these stolons actually grow across the surface of the food, right? So if we're thinking about this bread down here, all of the stuff that we see would be those stolons. Right, because they're on the surface of that food. Even over here, we would have stolons that are on the surface. All right. So we have stolons, but we're also going to have some things called rhizoids. And rhizoids are another specialized hyphae, but rhizoids go into the food. All right, so they actually go into the food to absorb the nutrients that are coming from that food. So this is one of the reasons why when you have something like moldy bread, you want to throw the whole thing of moldy bread away because, right, those rhizoids are actually in the food. So just cutting off the bad part doesn't actually ensure that you're getting all of the mold off, right? So we have stolons and rhizoids. Stolons are on the outside, rhizoids go inside. Now, we do have an interesting life cycle here. We're going to have both a sexual and asexual portion of that life cycle, right? And in the asexual reproductive portion, we're going to have sporangia. Remember, sporangia is going to be producing spores. So we have specialized sporangia called zygos, or that are going to be producing zygospores. So we have sporangia, they make spores, and those spores are called zygospores. So that is making that. So over here, we've got a couple things that we can look at. All right, so we're going to have our stolons, which are on the outside. Right, remember, rhizoids go inside. All right, and we're going to have the sporangia that is on the right off of the stolons that's going to be making those spores. All right, so that would be asexual reproduction right here. All right, so asexual reproduction, right, we have our sporangia that's on the stolons. They're making spores, right? They're landing, germinating, living their best life. But they can do sexual reproduction as well, right? But sexual reproduction is only going to happen when it is in no longer favorable conditions. So once unfavorable conditions come around, we will have sexual reproduction that starts. Now, we don't necessarily have male and female. We have positive and negative mating strands. All right, so we don't necessarily have a male female, but we do have positive and negative mating strands. And so what happens in this process, if we've reached these unfavorable conditions, we're going to have hyphae that produce gamtangia. And they're producing gamtangia. And gamtangia is a reproductive structure that contains haploid nuclei. All right, so we have haploid nuclei. So if we're looking down here, all right, we've got two mating strands here, right? So we'd have a positive and a negative. Right, and right here in the middle, they're going to be making that gamtangia, which we can see right there, right, which are then going to fuse those nuclei together in order to then make a diploid zygospore. Right, so then we have a diploid zygospore. Now, interestingly, those zygotes, that zygospore, right, that contains the zygote, is going to be able to remain dormant for months until that environment becomes favorable again. So these spores can last for a very, very long time, right? So they can last a long, long time. But once we reach those favorable conditions again, right, that zygospore is then going to germinate, right? It's going to make hyphae, it's going to make sporangia, which then makes spores, which then can go back into right onto that and making stolons and rhizoids and all of that. Right, so we have a zygospore that will actually produce hyphae, make a sporangium, make spores, right, haploid spores, that will then germinate into a new common mold. So that is the sexual reproduction portion of our common molds. And remember, common molds are in phylum Zygomycota. Right, we can remember Zygomycota because, right, they make zygospores. Make sense? I hope so. On to our third phylum that we're going to talk about, which are sac fungi. Sac fungi belong to phylum Ascomycota. And Ascomycota is going to contain more species than any other phylum of fungi. Right? So they contain the most fungi. And most of those sac fungi are going to be multicellular. Although it does contain yeasts. And remember, yeasts are unicellular. They are the unicellular fungi. So while most sac fungi are multicellular, yeasts are also a sac fungus. Right? And remember, right, from the last section of notes, yeast reproduce through budding. So they will have a slightly different reproductive cycle and life cycle compared to the multicellular sac fungi. But this is some examples of what sac fungi kind of look like, right? So we can have sac fungi that look like this. We can even have blue cheese. Blue cheese is made of a sac fungi, right? A sac fungus, and is used for the making of blue cheese, all right? But we've got a life cycle, all right? And we are still able to produce sexually and asexually, all right? So during asexual reproduction, we're going to have spores that are formed at the tips of hyphae, and those hyphae are called conidiophores. All right, so we have specialized hyphae, right? So we have specialized hyphae. There we go. That are called conidiophores, right? We're looking down here. So we have these conidiophores, they're specialized, and they are going to start producing these spores, or condia, all right? So these Condia, then, right, asexual reproduction, they then germinate into hyphae, which can then germinate into a new sac fungus, right? Remember, we're in phylum Ascomycota, is our conidiophores and condia. So that is asexual reproduction. That's happening over here. But we can have sexual reproduction that happens. And in sexual reproduction, we're going to have hyphae from opposite mating types that fuse, right, and make one nucleus, right, from each of the pairs of separate cells, all right? So in this sexual reproduction cycle, we're going to have a lot of terms, all right? So hyphae, right, we're going to have specialized hyphae called ascocarps, all right? So hyphae develop in reproductive structures called ascocarps. That's going to be this part up here in this fruiting body. We're going to have ascocarps. So that's hyphae that are going to develop in there, right? And these haploid cells are going to fuse while in that ascocarp, right? So we're going to have ascus, right? So we have two ascus here, which then fuse to make a zygote, right? So we are looking more specifically, right? We've got two nuclei that are then fusing to make that zygote. So that portion is our zygote right there, right? And that zygote is then going to start undergoing meiosis, right? So we're going to start replicating and replicating and replicating, right? Until we have a structure called an ascus, right? So it divides and divides until we have an ascus. And that ascus will then start producing ascospores, all right? And those ascospores will then germinate and develop into hyphae, which can do asexual reproduction, right? Or even go back through this path to become and make that, right, ascocarp in order for, right, these ascus and the zygote to be fertilized and all of that, all right? So we have, right, an ascocarp, right? They're going to develop a zygote, which then undergoes meiosis in order to become an ascus, and that ascus produces ascospores. Remember, we are in phylum Ascomycota. The last of our fungi phylum that we're going to dive into is Basidiomycota. These are the club fungi. And in Basidiomycota and our club fungi, this is where we find our mushrooms, right? So our mushrooms can be saprophytic, parasitic, or even mutualistic, all right? And mushrooms can look very diverse. They can look very diverse, right? So in this life cycle, the interesting thing to note is that club fungi rarely produce asexual spores. They want to produce sexually. They want a sexual life cycle. So they rarely reproduce asexually, right? And interestingly enough, they're going to spend most of their life cycle as a dikaryotic mycelia. Let's break that down. Remember, mycelia, right, from our last section is if we have our mushroom, right, we're going to have mycelia that are under, right, getting nutrients. So it's that branching of hyphae, so highly branched hyphae. So we're going to have this mycelia. And remember, we still have hyphae that's inside. But then we get to dikaryotic. Di means two, and karyotic essentially means nuclei, right? So it has to do with the genome. So we're going to essentially have two nuclei, which we can find right here. So we're going to have dikaryotic mycelia. So that means each of the cells in that hyphae has two nuclei associated with it. And periodically, they're going to reproduce sexually by forming basidocarps, all right? And basidocarps are essentially a type of fruiting body. Remember, the fruiting body would be that part. So they're going to make basidocarps, all right? And this happens because they're going to grow very, very rapidly through cell enlargement. So they're absorbing a lot of water, nutrients, and things of that nature. And that basidocarp is going to be made of basida. Remember, basida, we're still in hyphae. It's a specialized hyphae. So basida is a club-shaped hyphae that will produce spores. So our basidocarp is made of basida, which is a hyphae, specialized hyphae. And that basidocarp that's made of basida will make basidospores. All right. So Right up here, we have our Basedocarp specialized fruiting body that is made of specialized hyphae called Baseda, which is then going to produce Basedospores, because we are in phylum Basedomycota. So that is the life cycle of our club fungi. Now there is one other group of fungi that we can talk about, and they are in phylum Deuteromycota, right? And in this phylum, they really only share one trait, right? So they really only share one trait, and that is that sexual reproduction has never been observed in this group of fungi. So all of the organisms that are in phylum Deuteromycota have never had sexual reproduction been observed. It doesn't necessarily mean that it's never happened, but it's never been observed. There's never been the conditions that have dictated sexual reproduction to happen. And so this group is nicknamed the imperfect fungi. Right? So because they don't do sexual reproduction, they are considered imperfect fungi. Right? And so, right, Deuteromycota is also kind of like a junk drawer phylum because they really only share one characteristic. And so, right, might change. Organisms in this phylum might end up getting reclassified into one of the 4 major phyla of fungi. So we'll see. Now, we do have some more specific things, right, that we can talk about in specific types of fungi. Lichens are the first one. So a lichen is essentially a fungus that is in a symbiotic relationship, right? It's in a symbiotic relationship, right, of a fungus and an alga or other photosynthetic partner. So remember, alga is a protist, plant-like protist. So we're at a plant-like protist. Or other photosynthetic partner, that could be bacteria or even plants. All right. So this is a symbiotic relationship. And in this symbiotic relationship, we've got a couple things that happen. So we're going to have hyphae, right? So we are going to have a bunch of hyphae. And those hyphae are going to join with alga, right? And the hyphae essentially hold the alga where it's supposed to go, right? And they trade nutrients, right? So the fungus provides structure and habitat for the photosynthetic partner, and the photosynthetic partner gives food, right, from photosynthesis. So we have this symbiotic relationship where the fungus gives structure and the photosynthetic partner gives food, right? So we have structure and food that is getting passed back and forth. So that would be considered a lichen. Now, there are a ton of different things that can be considered lichens. There's over 25,000 different species that we consider lichens. But the interesting thing is that lichens are really only found in really harsh environments. So they're typically found in very harsh environments because they only need light, air, and minerals to grow. Because remember, they're getting their food from that photosynthetic partner. So they only need light, air, and minerals to grow, which means they often serve as a pioneer species. And if you don't remember, a pioneer species is essentially the first thing that comes back to an area after, right, a major disaster, right? So it conditions that area in order to make soil so that things can grow. So if we're thinking about an example, right, if we're on an island in Hawaii, we have a lovely volcano, right? Volcano erupts, right? And we have lava that goes everywhere. Lava kills, right, all of the plants and things that were there. So how do we actually get soil back, right, on a tropical place like Hawaii? We need a pioneer species. So lichens can be found a lot of different places in places like Hawaii where we have active volcanoes that are erupting. But interestingly enough, lichens are also very sensitive to airborne pollutants. So they're very sensitive to pollutants. And so they actually can serve as bioindicators as well, right? And so what can happen is if, right, we have a bunch of lichens and pollution comes in, those lichens are going to actually start dying. And in the process of them dying, we could then know that the air is not that great and there's a lot of pollutants. All right, so they are a bioindicator. So they're just very sensitive and responsive to changes in the environment. So they're a bioindicator. So over here would be an example of when a lichen actually starts changing color because of pollution in the air. So it starts turning this really weird red color. Right? For reference, this is what it should look like. Now, that's not the only kind of special structures and fungi relationships that we've got. We can also have mycorrhizae. And mycorrhizae are a mutualistic relationship between a fungus, let me change this, a fungus and a plant root. So we're looking specifically at a fungus and a plant root. So these is going to be multicellular. Right, so up here, lichens were typically unicellular. Right? So if we go up here, we have unicellular things. Here, we're going to have multicellular things. So we're looking specifically at plant roots. All right, and so what happens in this mutualistic relationship is we're going to have the fungus that absorbs and concentrates. Right, so we're absorbing and concentrating different minerals in the soil for that plant. Right, so this is going to help it reach extra water. It's going to help it find extra nutrients in the soil. It's going to help it get all the things it needs to survive. And in return for giving the plant all of these things, the fungus gets carbs and amino acids, right? Because remember, this is a plant, right? And that plant's going to be doing photosynthesis, so it's going to be having, right? It's going to be making carbohydrates, so it's going to send some carbohydrates down to the fungus, along with any amino acids that get made as well from their cellular processes. So we can have mycorrhizae that also do this. So this would be an example here, right, where we have the tip of a plant root, right, and all of that would be considered that fungus, so a mycorrhizae, right? And that's kind of what it would look like if you really zoomed in on it. Now we do have some beneficial fungi, and fungi are used for a lot of different things, one of which are medical uses, right? So we can use fungi for different medical things like making antibiotics, even making high blood pressure meds. We use them to control bleeding. We can treat migraines with them. And we can even make some medications that reduce organ rejection after transplantation. So there's a lot of fungi that have a lot of very good things that are used for medicine, specifically making medicines. But we can also obviously have fungi as food, so specifically our mushrooms, or in the production of food like yeast in breads. And we can use fungi for bioremediation. And bioremediation is where we actually take pollutants out of the soil. Right? So this goes back to our lichens and all of that kind of fun stuff. So we do have this bioremediation that can also happen with our fungi. But we can also have harmful fungi. So we can have fungi that are parasites, right? Parasites to humans and other organisms and crops for that nature, right? So some examples of a harmful fungi would be athlete's foot. Ringworm is also a fungus. Yeast infections, right? All of these things would be caused by a fungus. So this is a picture of ringworm. Right? We have here a fungal infection of some grapes. Right? And so we do have some bad fungi that can be harmful, right? Because they can be parasites. But that does end this section of notes, so make sure that you go back through all of these essential questions, and I'll see you later.